Hello everyone, this is Medical Gateway, and in this part we are going to study regarding the lifespan of RBCs. So the lifespan of red blood cells is 120 days, which is 4 months. So after 4 months, the red blood cells basically die. So why exactly the red blood cells die after 4 months, and what is the mechanism? So in order to understand, we have to understand first of all few things. That red blood cells doesn't have nucleus, mitochondria, or endoplasmic reticulum. So if a cell is not having these things, then how the cell is going to produce energy? But still, we know that RBCs can produce ATPs. So how do they produce ATPs without having the mitochondria? So they produce energy because of the cytoplasmic enzymes present in the red blood cells. The red blood cells doesn't have nucleus, mitochondria, and or endoplasmic reticulum, but they do have cytoplasmic enzymes within them. Okay, now, so what are the function of these cytoplasmic enzymes? These cytoplasmic enzymes basically maintain the cell membrane pliability of RBCs. So cell membrane pliability is very important. The cell membrane of the RBCs is in such a way that the RBCs have a biconcave disc structure, and the cell membrane is much more as compared to the content inside the RBCs. Therefore, this cell membrane and its pliability and its flexibility allow the red blood cells to squeeze through any small opening. Second one is the cytoplasmic enzymes help in maintaining the membrane transport to the cell membrane of red blood cells. Now, the membrane transport. The membrane transport is very necessary in RBCs because the oxygen has to pass the membrane and the carbon dioxide has to cross the membrane. So that's why cytoplasmic enzymes maintain this transport. Then cytoplasmic enzymes maintain ferrous form of iron. So iron has to be present or maintained in ferrous form, not the ferric form. Okay, so that is maintained by cytoplasmic enzymes. Then finally, the cytoplasmic enzymes prevent oxidation in the RBCs. So these are the four functions: maintaining cell pliability, and then maintaining the membrane transport, and then maintaining the ferrous form, not the ferric one, and then prevention of oxidation. Now, what will happen if these enzymes become less active? So after 120 days or with time, basically what is happening is that these enzymes which are performing these functions, these enzymes become less active. So when they will become less active, then they will not be able to maintain the cell membrane pliability. The membrane will become fragile. Now just imagine an RBC at 84 micrometer RBC, which has fragile membrane, is passing through a 3 micrometer splenic red pulp trabecular space. Definitely an 84 micrometer fragile membrane cannot pass through 3 micrometer space, so it will be destroyed while passing through the splenic trabecular spaces. Now, when they will be destroyed, so in the liver and the spleen, there are already present the macrophages. These macrophages will phagocytose the RBC, and specifically the hemoglobin present inside the red blood cells. Now, when the hemoglobin is phagocytized and the RBC is dead basically after 120 days, now what is going to happen with this hemoglobin when the macrophages eat them up? Now, as you know, hemoglobin is made up of heme and globin. So globin is the protein. So the globin part detaches, and heme is made up of iron and a porphyrin ring. Okay? So this iron is basically goes to the ferritin stores, or this iron goes to the formation of the new RBCs. So there are two things which can be done with this iron. Either it goes to the fat in stores, or this iron can be utilized to make new RBCs. And what is going to happen with this porphyrin? So this porphyrin will be converted into bilirubin, and that will be released in bile. So this is that bilirubin, which when the level of this bilirubin is raised, that is known as jaundice, the yellowish discoloration of skin. So this is bilirubin. Whenever there is increased breakdown of RBCs, increased amount of porphyrin is produced and increased amount of bilirubin is produced, which gives yellowish discoloration to the skin. So this is the regarding the topic lifespan of RBCs. The RBCs have 120 days of lifespan. And why they are being dying after 120 days, because the cytoplasmic enzymes which are maintaining all these functions becomes less active and the cell is finally destroyed. Okay, so this is it. See you in next part.